Hi, in this screencast we are going to discuss gene regulation. Gene regulation results in differential gene expression, leading to cell specialization. This just discusses the fact that although in every cell they have a complete copy of every gene, and although that may be true, not all of the genes are going to be expressed by every particular cell. So as we just said, every cell has a copy of every single gene, but does not express every single gene. Not all cells in my body produce, for instance, blue pigment, but the cells in my iris of my eyeball do. So the big questions are, how are genes turned on and off? And how do cells with the same genes, like I just gave you an example of, how do they differentiate to perform completely different yet very specialized functions? Gene expression, or the expression of each individual gene, is tightly regulated, and it's really complex, although we're not going to make it much harder than we need to at our level. Gene expression can be modified to counteract environmental changes that might be encountered, or they might just change to alter the function in the cell. Constitutive proteins are ones that are going to be actively expressed all the time. They are going to be such things as cellular structures that are a main component of all the cells. Inducible genes, however, are expressed only when their proteins are needed by the cell, when they are called to be made by the cell. For instance, as I gave you an example, the particular protein that acts as a pigment in the iris of an eye, or the proteins that are turned on to produce nails or hair fibers, are not going to be produced by liver cells in the body. Because transcription and translation are basically simultaneous in prokaryotic cells, there's little opportunity to regulate gene expression after transcription. So the control of genes in prokaryotes really has to be done by turning transcription on or off. It's the most efficient means of regulating gene expression in prokaryotes, and that's the level of transcription. It's slower, but it's greater savings of energy and resources. So as we just stated, prokaryotes conserve energy by only making the proteins that are needed, and especially in a rapidly changing environment, it allows them to be very efficient in what they need to get done. For example, E. coli, which lives in the digestive tract of organisms, including humans, they have to adapt quickly. What the organism, what the animal, what a human eats is what the E. coli get to use. So whatever comes down the GI tract is what E. coli must adapt to. Eukaryotes, on the other hand, are obviously multicellular, and they are much more complex. They have evolved to maintain homeostatic conditions, to regulate their body with growth and development through specialization is the big need or explanation for why gene regulation needs to occur. With specialization, each cell of a multicellular organism expresses only a small little fraction of the genes. For instance, humans have diploid human cells have 23 pairs of chromosomes, and only a small fraction of those genes are expressed by each individual cell. Different genes are needed at different parts in the life cycle. And after those times, then those genes can be turned off permanently, and continually responding to organisms' needs through homeostatic mechanisms. Cells of multicellular organisms continually turn certain genes off and on in response to signals from their environment. So gene regulation in eukaryotes is much more complex, but it's also much more needed. Greater complexity, greater number of points for control. So with prokaryotes, we really talked about only gene regulation taking place at transcription, whether or not transcription is going to take place. And that still is a main point in humans as well, but there's other points, and we're not going to look at all of these, or nor are you going to need all of these. But I'm going to repeat myself. Greater complexity in eukaryotes opens the door for greater numbers of points where gene regulation can be controlled. So again, with eukaryotes, we'll discuss the transcription level at which gene regulation can take place. Gene regulation or expression begins at the promoter, and we remember that from discussing DNA transcription into RNA. That's where the transcription is initiated. And basically it comes down to the quote-unquote decision as to whether or not specific genes on a DNA segment are going to be transcribed into RNA. And so there's two types of regulatory proteins, or in other words, are also called transcriptional factors. So again, whether or not transcription is going to take place. These two factors are going to be either a repressor or an activator. So in negative regulation, the presence of a repressor at a certain point will block further transcription. In positive regulation, the presence of what is called an activator will then signal transcription to continue. So in revisiting the transcription complex, the pretty complex collection of different proteins that are going to determine whether or not transcription proceeds or not. Some transcription factors are activators, remember, and some are actually repressors, things that decrease the expression of specific genes. Another place where gene regulation can take place is after transcription. Post-transcriptional control can involve RNA splicing. Remember, the splicing needs to take place basically to clean up the mRNA sequences or strands after transcription is done. And remember that the introns are going to be the junk DNA, the in-between stuff is going to be essentially spliced out of the functioning gene, away from the exons. But this can sometimes be done in a variable way. Different processing of those exons might create similar but slightly different proteins. An additional place where we can regulate the expression of genes can be controlling the progression of translation or the inhibition of translation. For instance, we can block initiation of transition by specific regulatory proteins that might attach to the end of an mRNA sequence and thus actually block translation from even occurring. And also, the cell limits the lifetimes of normal proteins by selective degradation or breaking down of those proteins. So many proteins, including ones we've talked about like cyclins that were involved in regulating the cell cycle, they could be relatively short-lived. They could be broken down because they are not needed anymore. Also, just protein processing. How exactly tertiary structure of proteins, the folding and cleaving and the addition of smaller groups within the huge polymer are added. The processing, the making, the folding, and also the breaking down and recycling of the protein. All are points of possible gene regulation. Just a summary for you to review. Some of these we talked about, some of them we did not. Just note that in eukaryotes there are multiple and many access points at which gene regulation can and does take place. And it's really important because of the complexity and all the specialization and the differentiation that takes place from one cell to the next in eukaryotes.